Welcome back to continuation of another class of RNA biology. So we were here in the last class that we reached up to LUCA, that is last unique common ancestor. And this LUCA has acted as the fundamental life form which eventually evolved into unicellular and multicellular organism. Now, let us think, do we have any evidences for the RNA world? We have been saying about RNA world a lot, but do we have any evidence in the modern world? So, do we have any relics? We have already mentioned about two of them. One is existence of ribozymes and also the presence of ribosomes. More than that, do we have any real RNA nuclei left out in the living organisms of the modern world? And some of them are listed here. The classic example is the ATP. All of you would have heard about ATP, that is adenosine triphosphate, which is the major source of energy for almost all biological reaction. And I'm not forgetting about GTP because many signaling events rely on the GTP, which is guanosine triphosphate. ATP stands for adenosine triphosphate. This molecule have got an adenosine nuclei followed by 3 phosphate groups. So this third phosphate is the one which is used for energy yielding reactions. And this is nothing but a monomer of RNA or DNA. So this is ATP is a monomer of RNA, not DNA. Because we will come to the difference of the ribose sugar much later. And then another example is S-adenosyl methionine. And then comes flavin adenine dinucleotide. Some of them we classify them as vitamins. Vitamins are essential components of several protein enzymes in the living system. And then you can have niacinamide, which is, we also call it as niacin, which is coming under the B family of vitamins. Then you have the classic coenzyme A. And then we have got the biotin. So these are all molecules, we call them as vitamins, etc. But they are all part of the RNA world, which staying back even today in the modern life forms, including humans. Now, let us think how an RNA monomer is formed. We know the ribose and the base plus the phosphate adds to a nucleotide. So, how does this happen? The nitrogenous base can be formed from HCN or hydrocyanic acid, and also the phosphate, phosphate can come from the rock weathering, and ribose sugar can be formed by the Formose reaction. And you can also have purines and pyrimidines that is formed from the inorganic material. And this can eventually combine, and we call them as nucleosides. And this eventually can combine with phosphate that is formed from the rock weathering. And we call it as phosphorylation. In biology, any molecule that is added with a phosphate group, we usually call them as phosphorylation. So the nucleosides, which is nothing but nitrogen base plus sugar, that is the pentose sugar, when added with phosphate group, we call that phosphorylated nucleosides, that is also called as activated nucleotides, which can undergo the so-called polymerization reaction. Polymerization happens between two nucleotides, and which direction we can discuss later. But this assembly of this monomer, the so-called nucleotides, can give rise to an extended molecule called RNA. Now, RNA first is a view of the RNA world hypothesis, and it is basically considered as a molecular biologist's dream because it is very easy to explain how the life or the molecular structure or the cell biology of a living organism evolved over the years. But it also triggers a prebiotic chemist's nightmare. So we can understand that RNA first molecule concept is a molecular biologist's dream because it is much easy to explain. Whereas when you talk about the chemistry, it is also a nightmare because of the conditions in which the prebiotic world existed. So let us look back into the RNA world again. To make an RNA, what you actually need, we know that it needs sugar and bases. But when the prebiotic surroundings are able to synthesize sugar or base in the proportion and in the quantity, were they able to produce? This is a big question because although you can produce one molecule or two molecule, that is not going to trigger the production of the so-called RNA molecule capable of giving rise to life forms. So what we should understand that this should have been a feasible reaction and a preferred reaction and also a sustainable reaction. And the answer is yes. Despite all the odds, the conditions of the prebiotic world was very much in favor of not only production of sugars and bases, but they also get selectively stabilized and preferred. Let us see how can we go about this further. If you look into this left panel top, few molecules are kept in box. That 3' 5' phosphate, and you also have 2' 5' pyrophosphate, 2' 2' polyphosphate, 3' 3' alkyl phosphate, and so on and so forth. And if you see left hand side down, you also have beta D ribofuranos, and you have got alpha L lixopyranose. What this D stands for, dextrorotatory, that is the chemistry, this optical activity of organic molecules, and L stands for levorotatory. Those who are studied chemistry will know that optical activity is a property of various chemical molecules which has the ability to rotate the plane of polarization of a plane polarized right while in solution. I will not go into the detail because it is part of chemistry, but remember, all amino acids in a living system are L forms, whereas the carbohydrates are D forms in all living organisms. Exceptions are there, but by and large, that's how it is preferred. So you are talking about different types of sugars in the left hand panel down here. And at right side, you can see adenine and guanine. We know they are purines, and you also have diaminopurine, hypoxanthine, xanthine, isoguanine, etc. etc. And remember, of all these purines that are formed, adenine and guanine are only circled, and you can guess why it is so. And in bottom, you have a list of pyrimidines, that is cytosine and uracil are given in circle, whereas you also have diaminopyrimidine, dihydrouracil, orotic acid, etc. And so what this shows that in prebiotic world, due to the lot of physical and thermal activity, a lot of molecules could have been formed and existed. However, not all of them are recruited into the RNA structure. Some of them are recruited. What could be that reason? And we should not forget that this hypoxanthine and xanthine, these are all pigmentation molecules even in flowers, even today, because a plant produces this molecule by chemical reactions to give coloration to various parts of its flower, etc. So these molecules are still there, but they are not part of the RNA molecule. So like you can see here in this slide, whichever mentioned in box, they all have given rise to becoming eventually part of the RNA. So looking further into the RNA world, do we have evidence for sugar? And is it possible that prebiotically sugars can be synthesized? And the answer is yes. And the reaction is called Formose reaction. And it was discovered by Alexander Butlerov in 1861. That Formose reaction is pretty simple. It is, although it is a lengthy process, the starting material is formaldehyde. What is the chemical formula? CH2O. And formaldehyde can combine, and formaldehyde can be spontaneously formed in the prebiotic world, and they combine and give rise to glycoaldehyde, and they again combine with formaldehyde and give rise to intermediate compound. And remember, they are reversible reaction. That means reaction can go in either way. Eventually, you can end up getting glycoaldehyde that can react with a bunch of tetrose, pentose and hexose sugars, and you end up getting a diverse sugar molecule. And remember, the ribose sugar is the one which is preferred and recruited into the RNA molecule. Reasons can be many, and which we will discuss one by one. So let us quickly think, how do the nucleotides polymerize? Polymerization, we know that the assembly of the monomers, which is the nucleotides. And once they polymerize, the end product has to be a polymer called RNA. So it starts with a phosphodiester linkage. What is a phosphodiester linkage? Phosphodiester linkage basically is formed between a sugar and phosphate group, which continues over downstream chain. It is formed by a condensation reaction. A condensation reaction is what? One hydrogen molecule and a hydroxyl molecule react together and one water molecule is formed, H plus OH. So one molecule is H and another molecule is OH. Together participate in release of an H2O. One water molecule will be released, and here in this formation the phosphate group and the sugar participate, the pentose sugar, ribose sugar participate in this condensation reaction, and it continues. And the sugar, like I told you in the previous class, it has got 5 carbon. Carbon number 1, carbon number 2, carbon number 3, carbon number 4, carbon number 5. So the phosphate reacts with the carbon number 5, and the subsequent downstream, the 3' carbon participate in the downstream, and the 5' end reacts with the phosphate group. Like that the chain continues. And so we should understand the bond formed between the phosphate group of one nucleotide and the hydroxyl group of the sugar component, and you end up getting a release of water molecule. H2O will be released, and this is called the fundamental backbone or the basic backbone of a RNA molecule. So the nucleotides involved contain the ribose, and this phosphate reacting with the ribose, which already containing a nitrogen base, which we call it as nucleoside. And this polymer now we call it as nucleotide polymer or RNA or ribonucleic acid. If the nucleotides involved that contains the sugar deoxyribose, then the polymer is called DNA. So like I told you, the ribose sugar have got 5 carbon atom, and the second and third carbon is very important. In DNA, the second carbon do not have an oxygen, whereas the third one have got a hydroxyl group. Second carbon has a OH group in the case of RNA, but in the case of DNA, the second carbon has got only the H. Oxygen is missing. That is why we use the word deoxy. Whereas the 3' of both DNA and the RNA, the 3' carbon has got OH group, which participate in the polymerization reaction. So the 3' and the 5' is important molecule. 3' and 5' carbon of the pentose sugar or the ribose sugar is very important for the polymerization reaction. So this is what you can see, the DNA or the RNA that has got a typical sugar phosphate backbone, as you can see here, and the nitrogenous base is capable of pairing with another nitrogen base pair of another RNA or another DNA, and this adenine nitrogen base pairs with the thymine, and whereas the guanine nitrogen base pairs with the cytosine. So adenine pairing with thymine or adenine pairing with uracil. Remember, RNA prefers uracil over thymine, although they are molecularly very close by. We will see them more in detail. A always pairs with T or U in a double bond, whereas G pairs with C. This is the short form. A is the short form of adenine, T is the short form of thymine, or U for uracil, and C for cytosine, and G for guanosine. So G to C pairing is always a triple bond. A to T or A to C is always a double bond, as you can see here. Remember, this is shown with respect to DNA, but RNA also have the ability to form such pairing. So the RNA's sugar phosphate backbone is very much similar to that of the DNA, but only difference is the sugar has got OH group in the 2', rest everything remains the same. And there is also a directionality. When you talk about the RNA or DNA, for a given strand, we always refer from 5' to 3' end. That is the 5' carbon to the 3' carbon end. Say, if I want to say left side and right side, if I am giving you a RNA which is like a ribbon, it is lying in front of it, has got a left end and a right end. So the left end will always be 5' and the right end always will be 3'. We will come to more discussion about this much later. Time being, you just need to know this much. So if you think about the prebiotic synthesis of sugars by Formose reaction, we have to understand lot of such molecules are formed by the Formose reaction, because Formose reaction do not favor a pentose sugar ribose. It can favor lot of molecules, and we have seen many of them are bidirectional. So what is so much great about this pentose sugar ribose? Where does the selectivity come from? Who is selecting? Is there someone sitting and selecting, okay, this molecule is important for RNA? That is something which we have to bother. And this is a complex slide, but don't worry about the molecular structure in detail. What we should understand is there is a mineral that is called borate mineral that is very important in preferring selectivity. That means if you see certain molecules, borate influences diastereoselectivity, whether D is to be preferred over L or a pentose sugar is to be preferred over hexose sugar or any other more complex structure, is all decided by the borate mineral that is present. Borate can also influence regioselectivity, and borate can inhibit the propagation to a particular form. Say a pentose sugar should not become hexose sugar, that can be inhibited, and borate can also stabilize some specific type of pentose sugar, and borate along with various other molecules can facilitate the selective accumulation of a given type of sugar. This doesn't mean that borate will prevent the formation of other sugar molecule, but presence of borate minerals can enhance the availability of a given type of sugar. It's like if you see a given fruit item is available in a fruit market. In mango season, if you go, you will see every vendor will have only mango. So if you go for buying fruit, there is a good chance that you end up returning home with mangoes. Same way, if you have only a given type of pentose sugar, then there is a good chance that pentose sugar will be incorporated in meaningful or without any meaning into some or the other molecules. And credit goes to the so-called selectivity provided by the borate mineral. And let us think in RNA world, do we have evidence for bases that are being formed? And you can see hydrogen cyanide, that two hydrogen cyanide can form a dimer or hydrocyanic acid, and it can keep on combining and it can give rise to a sequential reaction event, and in the end of the day you end up getting adenine, which is nothing but a pentamer of hydrogen cyanide. Hydrogen cyanide or hydrocyanic acid, which is also present in lot of tuber plants like such as tapioca. It has got, like, that's why if you are cooking some of this underground tuber plant, you put them in water for a long time to get rid of hydrocyanic acid or hydrogen cyanide. Hydrogen cyanide, remember, at high concentration can be very toxic. So otherwise people end up getting headache, etc. So hydrogen cyanide is a very easily available molecule. All it has hydrogen, carbon and nitrogen. So this is abundantly present in the prebiotic world. So as you can see here, xanthine can be formed, hypoxanthine can be formed, and also adenine can be formed. Similarly, other purine, that is the guanine, also can be easily formed by similar chemistry. So purines can be formed as a polymer of hydrogen cyanide. And let us look into the structure of the purines. Adenine and guanine, they are very much similar in structure, except that an amino group is in the adenine, and whereas the amino group is changed in its position in the case of guanine, and the place where the amino group is there in the adenine, now you have got an oxygen, whereas rest of the chains and parts are not very drastically different. They are more or less the same, and they are coming under the category called purines. Now let us look into pyrimidines. Pyrimidines are of two types, thymine and cytosine. In the DNA, whereas uracil and cytosine is in the RNA. If you look closely into the structure, they are also very much similar. That the thymine has got a methyl group, whereas cytosine has got a amino group. If you look closely, thymine and cytosine, and then I have mentioned here about the uracil. Uracil and thymine are not present together. In DNA you have got thymine, whereas RNA you have got uracil. Both cannot exist in one molecule. Either any one of them, only it can exist in the entire species of RNA species or DNA species molecules. And cytosine is somewhere in between. Interestingly, what you have to see is uracil and thymine are one and the same except for one methyl group. We can also call uracil as methylated uracil, that is thymine. So if you take uracil and add a methyl group, it becomes thymine. And we should also understand in higher order epigenetics, you also study the cytosine also can undergo similar methylation, and we call it as methylated cytosine. So understand the uracil has undergone methylation to become thymine, which eventually become part of the DNA. Otherwise, there is no difference between the uracil and thymine. Now, if you think about the prebiotic synthesis of pyrimidines, it is little difficult than purine, but it is definitely possible. And the ribose and the base can combine to give rise to nucleoside, and the phosphorylation of these nucleosides give rise to nucleotides. So what are we proving here? We are proving that we can easily have all the monomers of the RNA and which can assemble together and become eventually an RNA molecule. So now let us quickly go into Chargaff's theory. Chargaff is a scientist who discovered by chemical means that the number of adenine is equal to number of thymine on a DNA, and the number of cytosine is equal to number of guanine. This kind of gave clue probably A have something to do with the T or C have something to do with the G, and this gave the hypothesis that they might be pairing. This was very useful for the discovery of structure of the DNA. And he found that the bases must be relevant to its matching pair, else it is impossible to have a equivalent number between A's and T's and G's and C's. This is what you can see from the picture. Thymine always pair with adenine, cytosine always pair with guanine. And remember, in the case of RNA, thymine need not be thymine, it is the uracil, but the pairing rest of them remains the same. And in 1953, Watson and Crick discovered the structure of the DNA, and this emphasized and this established this pairing concept, and this is very important. You may wonder why talking about DNA in an RNA biology class, because the discovery of DNA has proven the ability of these nucleotides or the nucleic acids to polymerize and also pair, because DNA is a pair of two strands and they form a DNA double helix, and they depended on the results, extracrystallography results from Maurice Wilkin, Rosalind Franklin, etc., and they discovered the structure of the DNA. So this is a picture of Watson and Crick. And let us think, what's wrong if DNA has got lots of such features, pairing, structure, etc. Why not think DNA as the starting material? Why RNA? What is so unique about it? As we have already discussed, the DNA seems like a great suspect of being the first polymer to reproduce itself. But is it genuinely true? Because it poses a big problem. What is that problem? Problem is DNA is too simple and extremely stable to act as a template and to act as a catalyst and to fuel itself to form a new copy. Like we already discussed that if any molecule, any life form has to have any success, it should be able to make a copy of itself. If it did not make a copy of itself, it will disappear from the face of Earth. So DNA is very stable, but is it active? No. Can it be more dynamic? The answer is no. Then how can it perform a function? And The answer shows that it simply may not be a good candidate to act as a early biological molecule, and especially when it comes to self-replication. So in fact, never ever DNA has been shown to act as a catalyst in a laboratory. Like various approaches have been taken to prove whether DNA can act as a catalyst. No, DNA never could perform any biological reaction on its own. Remember, on its own. Here we are talking about without any help from other outside, which means without any external energy source, DNA is very unlikely to be able to sustainably make a survival on its own, or in this case, it is self-replication. And the problem with DNA that DNA doesn't just replicate, then what it could be doing. We will see that more in detail, that why DNA is important and what is the contribution of DNA, what could it be so special about the DNA, and why the so-called RNA world eventually turned into DNA, and what is the benefit it was getting. Those things need to be discussed more in detail, and we will continue with that in the next class. Thank you.